Hello everyone. Today we'll be furthering our discussion of microbial metabolism and completing Chapter 8, Part 2. As a short review, we discussed metabolism in the previous chapter. Metabolism is the combined effect of catabolism and anabolism, which are two processes that either build up or break down components within the cell. Little memorization tip that I once used is catabolism has the word cat in it, and cats tend to be kind of destructive forces in nature. And so if you would like to think of it that way, you can think that catabolism is the breakdown of things, and anabolism would be the building up of things. And so we can see how these metabolism is made up of catabolism and anabolism. And how catabolism breaks down nutrients in order to release energy that was trapped in those compounds. And that energy can be used in anabolism, which is the building up of complex molecules, which requires the addition of energy. And as you'll recall, that energy is usually given from a compound called ATP, adenosine triphosphate. And when it is in the ATP form, it has a stored certain amount of energy in that triphosphate bond. And through the breaking of that or snapping of that bond, we can release energy. And in the process, we create ADP and inorganic phosphate, or in other words, a phosphate that is not attached to an organic compound. It is just free floating in the cell. Moreover, these chemical reactions that take place in metabolism require a process of activation energy. In this reaction coordinate, we can see progress of the reaction on the x-axis and the amount of free energy on the y-axis. And it's similar to the analogy of sliding down a hill, where the reactants on the left can go downhill towards products because the products would be lower in free energy. And that is the case with sliding down a hill, where you're closer to the gravitational pull. When you go from a higher place to a lower place, you're reducing the amount of free energy in that system. However, if all reactants were able to start from a place at the top of a hill, then they wouldn't be reactants anymore. Everything would be the products of that reaction. So the reason why we have reactants, or molecules that are in this beginning state in the first place, is because they have not reached the top of the hill in order to make it to the other side. And this top of the hill is the amount of free energy required to make a transitional state. This transitional state is a state that's in between the state of the reactants and the state of the products. In order to reach that transitional state, the person holding the sled has to walk to the top of the hill. And so this is the amount of activation energy required, written E sub A. So enzymes make these reactions occur faster by doing one thing only. They reduce the activation energy. In a sense, they provide a shortcut in order to go down the hill and make products. With microbes on Earth, one of the most preferred molecules for storing energy are carbohydrates. So most microbes obtain most of their energy by breaking down carbohydrates, and this is usually glucose. And this occurs via one of two pathways. It can occur through cellular respiration, as does with us humans and all animals, typically, and can also occur through fermentation. The end goal, though, is to oxidize glucose to generate ATP. To oxidize glucose, in layman's terms, is to decrease the amount of hydrogen bonds and carbon-to-carbon bonds and increase the amount of carbon to oxygen bonds. So the ultimate terminal product here would be CO2, or molecules that have a higher ratio of oxygen to carbon. In this process, we produce ATP. Now, a lot of these processes, they take a while to wrap one's head around them. And it's hard to be able to know what's important here. So I am going to say at the get-go that when you're looking at these processes, I want you to pay attention to certain things. One thing I want you to ask yourself is, where is the carbon going? Okay, so you're going to follow the carbon trail. I want you to notice what molecules are being built or degraded and how many carbons these molecules have. So you'll typically have a starting material that has a scaffold of carbons to it, like glucose has 6 carbons. And as that's degraded, you're going to notice that matter is not created nor destroyed in this process. So all of those carbons end up into different molecules along the way. The next thing you want to observe is, where's the energy going? So we have these molecules that store energy in their bonds, and as these bonds are broken or made, energy is released or stored. And so you need to pay attention to the ATP and ADP levels when this occurs. So ATP is spent in making complex molecules, and it's going to be harvested in the degradation of molecules. Finally, I want you to note where the electrons are going. So in these processes, we're separating out the concept of electrons and energy. We tend to think of electrons as energy because we have an electrified grid, and that has a electric potential. That is, one end is more negatively charged, one end is more positively charged, and the electrons will flow from the negatively charged to the more positively charged end. But that's not what's going on here. We have two separate things. We have energy that's stored up in bonds, and we have electrons that belong to atoms and molecules. So we'll see how those differences play out as we move forward, but keep track of those 3 things: carbon, energy, and electrons. Okay, so let's begin with glycolysis. Glycolysis is the breaking down of glucose. We've seen lysis before, which means to break apart. Glyco means sugar. And specifically, this is the sugar molecule glucose. So this is what the metabolic pathway for cellular respiration and fermentation look like. These are essentially 2 different ways of breaking down glucose, both of which start with a process called glycolysis. So let's start with cellular respiration. Cellular respiration begins with glucose, which goes through glycolysis, pyruvate oxidation, the Krebs cycle, and finally the electron transport chain. In cellular respiration, electrons are collected from glucose and its derivatives and uses their energy to generate ATP. It does so by using 2 molecules called FADH2 and NADH. And you can think of them as electron carriers. So they can move electrons from one molecule to another. When they are in their forms called FADH2 and NADH, they have the extra electrons full. So this is when they're on the job, so to speak. They have complicated names like nicotinamide adenine dinucleotide or flavin adenine dinucleotide. But for the purposes of this class, we can just refer to them as NAD or NADH and FAD and FADH2. You don't have to memorize these chemical structures. I put them here as just for your information and to help you memorize which state carries the electrons and which state does not. For NAD plus and NADH, it's a little bit more apparent because the name NAD plus hints that there's a lack of electrons because electrons are negatively charged, and we can see this molecule has a positive charge to it. That positive charge is because this nitrogen here has too many bonds, and so therefore it ends up being deficient in electrons. For FAD and FADH2, what occurs with this molecule is when it gains two hydrogens, it actually also picks up 2 electrons. So if you see FAD with the H2 there, you know that those hydrogens came with those electrons, and that charges this FAD. Molecules that are tend to be short in electrons, they're often called the oxidized form, and that's because in nature, oxygen really likes to take electrons from others. So you'll often see this as referred to as oxidized. Reduced forms in molecules, and there'll be more of these that we talk about in the future. They're molecules that tend to have a higher amount of electrons in this form. And because electrons are a negative value, we say that they're reduced here. So remember in the past we talked about reactive oxygen species. They're reactive and they're toxic because they go around snatching electrons from other molecules in the cell, thereby oxidizing them. This image is a 10,000-foot view of cellular respiration. We're going to get into greater detail for each one of these processes, but this shows that process of cellular respiration that I outlined. Here, we see glycolysis occur. Glycolysis occurs in the cytoplasm of the cell. And eventually, we get through pyruvate oxidation, which occurs here. And pyruvate oxidation makes acetyl-CoA. And that acetyl-CoA can travel into the mitochondria. Remember, the mitochondria was that organelle that is thought to be derived from bacteria long ago through a process called endosymbiosis. And that bacteria was thought to be capable of cellular respiration. So this continues the process of cellular respiration inside of the mitochondria. That compound that we made, acetyl-CoA, remember this is following the carbon trail, that acetyl-CoA goes into a process called the Krebs cycle, which is a series of metabolic steps. All of these processes, by the way, are controlled by enzymes that drive these reactions forward, right? Because reactions are rather slow, typically in biology, without the use of an enzyme, which reduces that activation energy. That acetyl-CoA enters into the mitochondria and continues through this metabolic pathway called the Krebs cycle. Eventually, components that are made up in the Krebs cycle go through the electron transport chain. And through the electron transport chain, even more ATP is created. We run through this cycle again. Let's start with glycolysis. We see that we have to use up a little ATP to prime this system. So we're actually losing energy in the beginning, but then later in glycolysis, we gain some ATP back. And then we get to pyruvate. Pyruvate becomes oxidized to make acetyl-CoA. And in this process, we gain some more ATP and another molecule called GTP, which is an analog of ATP. But what we're really doing here is we're making a bunch of these electron carriers, NADH and FADH2. And that we'll see is very useful in the electron transport chain. For the exam, you should know the starting material of glycolysis. In this example, it would be glucose. And in pyruvate oxidation, it's going to be pyruvate, right? You're also going to need the starting material in Krebs cycle, again following the carbon trail, which is acetyl-CoA, and for the electron transport chain, which are your electron carriers. You're going to need to know how much ATP is produced, a sum total of ATP in glycolysis and in the Krebs cycle and the electron transport chain in the total, as well as how much FADH2 and NADH is created from each. I know that seems like a lot, but we'll get through it. Let's begin with glycolysis. Glycolysis has a net total of 2 NADH electron carriers created and 2 ATP being created. Glycolysis leads to the production of 2 pyruvic acids, also known as pyruvates. The reason for that is glycolysis, remember, is the splitting or breaking down of glucose. And glucose is a 6-carbon molecule, and it becomes 2 3-carbon molecules. In pyruvate oxidation, it's a one-step process that takes your 2 pyruvic acids, which are your 2 3-carbon molecules, and for each one you get an NADH. So since there are 2 of them, you get 2 electron carriers, 2 NADHs. Remember, the term oxidation means that you're increasing the amount of oxygens that are attached to it, or in some cases you're removing electrons. So here we can see that for each pyruvic acid that goes through, we create 1 carbon dioxide for a total of 2 carbon dioxides. And out comes at the other end 2 acetyl-CoAs. And the acetyl part is a 2-carbon molecule. So there are 2 2-carbon molecules because they were once pyruvic acids that lost 1 of their carbon molecules. So they went from a 3-carbon molecule to a 2-carbon, and then the extra 2 carbons didn't disappear. We just created carbon dioxide out of them.
Checkpoint 1. How many pyruvic acids are produced from glycolysis, and then how many acetyl-CoAs are produced in the following pyruvate oxidations?
So now that we've covered glycolysis and pyruvate oxidation, we're ready to continue following the carbon through the process of cellular respiration. The acetyl-CoAs that have been created are allowed to enter into the mitochondria. Within the mitochondria, these acetyl-CoAs are destined through cellular respiration to enter the Krebs cycle. The Krebs cycle is called a cycle because the final product can react with a new acetyl-CoA to make the first intermediate compound, citric acid, to continue this process. So the citric acid eventually becomes oxaloacetate, which then can react with another acetyl-CoA to regenerate another citric acid. So, in this process, we take an acetyl-CoA and another acetyl-CoA, and which is the 2 products from pyruvate oxidation, and we create a bunch of NADH and FADH2. We also create GTP, which, as I hinted before, GTP can be thought of as basically an ATP. It serves the same function. It's a very similar compound. And completing this cellular respiration, we eventually will end up with 4 CO2s. So if we were to follow the carbon, how much carbon went in is acetyl-CoA? Hopefully you said 4 carbon molecules. Two carbon molecules that are fully degraded eventually will become 4 CO2s. There's 4 carbons going in and 4 carbons coming out. But can you remember the purpose of NADH and FADH2? In other words, what are they gathering from acetyl-CoA in this process? We can see here that a little bit of energy is gathered in the form of GTP, but our main compounds that are being made are these electron carriers. So we're actually gathering the electrons from acetyl-CoA. So this is far and away where most of the electrons are harvested. We get a grand total of 6 NADH and 2 FADH here. As we can see, in glycolysis, 2 NADHs are produced. In pyruvate, each pyruvate produces an NADH, so we get 2 more NADHs out of this. But the Krebs cycle, we collect 6 NADH and 2 FADH2s. And all of those electrons are consumed in the electron transport chain. Here is a diagram or a figure of the electron transport chain. Before we continue, you should be able to answer these questions of where is this all happening in the cell? What organelle did you learn that was the space where ATP is made? Hopefully you said the mitochondria. And do prokaryotes have a mitochondria? No, prokaryotes don't have a mitochondria. They typically don't have membrane-bound organelles, but they do complete cellular respiration, many of them, along their inner membrane. And if you look at this image, it has the appearance of a bilipid membrane. We can see that this is where the site of ATP is produced. If we start from left to right, we can see the general process. We have our electron carriers, NADH and FADH2, and they are regenerated into NAD plus and FAD. However, their electrons are donated to the electron transport chain, either through these complexes such as NADH dehydrogenase, or through these membrane-bound electron carriers like quinone and ubiquinone and compounds like that. We won't get into the details of memorizing each one of these complexes, but you can see it creates a chain of electrons, and these electrons are used in order to pump protons across this membrane to this inner membrane space. Take a moment and locate where oxygen is on this electron transport chain, and let's see if we can make sense of what the role of oxygen is in this system. We can see here at cytochrome oxidase complex that we can take an oxygen molecule. This reaction is balanced, so it writes it as a half an oxygen molecule in order to simplify things. But it reacts with the electrons. Remember, oxygen loves to take electrons, and a couple hydrogens, and it produces water. And that is the major role of oxygen. We call it the terminal electron acceptor in cellular respiration. So imagine when you're breathing in and you're taking in oxygen, right? And you're also breathing out CO2. This is the process of cellular respiration. You're breathing out CO2, which is actually the byproducts of cellular respiration. As we saw, the carbon eventually becomes carbon dioxide from glucose and other molecules as well. And your oxygen reacts with the electrons in the electron transport chain and two protons in order to create water. And what is all this process for? Well, eventually it is used to produce ATP, and it produces a massive quantity of ATP. We're looking at 34 ATP from the one glucose molecule alone. Let's go back to an analysis between bacterium or prokaryotes and mitochondrion. Here we can see that prokaryotes have their electron transport chains occur on the homologue of the mitochondrion's inner membrane, which would be their inner membrane, their plasma membrane. In eukaryotes, the cellular respiration occurs across the inner mitochondrial membrane, as we can see in the second figure here. And it's made up of a very complex folded, they call these invaginations of the inner membrane. And what that does is that increases the surface area so that the mitochondria can do more and more of this cellular respiration than what would be defined as if it just had a round, contiguous inner membrane. So the protons that are pumped across that membrane in bacterium, they build up in the periplasmic space here. And in mitochondria, they occur in what we just call the intermembrane space.
Checkpoint 2. In which stage of cellular respiration are the majority of electrons harvested for later use? Hint: that later use is where ATP synthesis uses those harvested electrons.
So now, I want you to imagine what would happen if there was no oxygen waiting at the end. If we were to have our electron carriers in the beginning here, and they donated their electrons, and we can see that those electrons travel through the chain and end up with oxygen in the end. What would happen if you didn't have enough oxygen present or no oxygen present? Naturally, this chain would get backed up, and this process could not function because the electrons would have nowhere to go, and so you could not get a proton gradient or a higher concentration of protons on the other side within the intermembrane space. These protons are eventually all gathered and they run through this ATP synthase. You can kind of think of it in the way a dam's turbines might work. This charges this turbine here, and this turbine turns, and that turning energy is used to convert ADP, inorganic phosphate, into ATP within the mitochondrial matrix. So if we were to remove our oxygen here, this chain would get backed up and cellular respiration would come to a grinding halt. There are mechanisms to circumvent this problem, and we can see that in this example here, glycolysis, if we use cellular respiration, we have this process of regeneration. So this NADH in the electron transport chain can be regenerated into NAD plus after it donates its electrons to the electron transport chain. And that NAD plus can therefore be used again in glycolysis. So theoretically, and this does play out in real life, you could exist on more meager forms of ATP production. So remember, we saw that 34 ATP were produced per glucose there. We could exist through a process called fermentation, but we have to have a way of regenerating our NAD+. So without any oxygen in cellular respiration, we don't capture those electrons, and therefore we cannot dispose of our electrons from our electron carriers in glycolysis and the citric acid cycle. And so therefore this regeneration process cannot occur. So glycolysis, again, it's wonderful because we get 2 ATP, or sorry, cellular respiration is wonderful because we get 2 ATP from glycolysis, and we get 36 ATP from the rest of the process. But without the electron transport chain, it is a futile to attempt to do pyruvate oxidation, the citric acid cycle, and electron transport. Instead, fermentation delivers electrons from glucose to an organic molecule, so not oxygen. And remember, oxygen was called the terminal electron acceptor in cellular respiration. And the definition of fermentation is that this is a catabolic process in which uses a organic terminal electron acceptor. And in this process of dumping the electrons from your electron carriers to other organic molecules, we can regenerate our ATP, or sorry, our NAD plus, and reuse that for more glycolysis. And the way this looks is we have glycolysis to produce 2 NADHs and 2 ATPs. And we have 2 pyruvic acids. And in fermentation, we take the pyruvic acids and our NADHs, and we donate them to organic compounds to regenerate our NAD plus and continue doing more glycolysis. This often leads to the production of 2 CO2s and 2 ethanols. Remember, if we follow the carbon trail, we have a 1 molecule with 6 carbons. We create 2 molecules of 3 carbons, which is still 6. And ethanol is a 2 carbon molecule. So we have two carbon molecules, which is 4. So to get 6, we add back our CO2s, 2 CO2s, so 2 plus 4 is 6. All of the carbon can be tracked through this process. So I'm sure that you've heard of fermentation to produce ethanol. There are other methods of doing fermentation which use different terminal electron acceptors, but they all share a similar trait in that they produce about 2 ATP from glycolysis, which is not favorable, but if you have no other options, then why not? I mean, there are many environments which do not have a plentiful amount of oxygen. In fact, this planet as a whole was very oxygen poor long ago. We had what we called a reduced environment or atmosphere that was not rich in oxygen. And it wasn't until the development of oxygenic photosynthesis, or in other words, like plants, algae, cyanobacteria, that produced oxygen by harvesting the power of the sun, that we had an oxygen-rich atmosphere. So this was kind of more of the normal than kind of just trying to scratch out an existence in other harsh environments. So, there are pros and cons to this. While fermentation produces many fewer ATP than cellular respiration, it can happen very quickly. It's a lot less work in the end, and there's a lot shorter process, so it happens very quickly, and it can be done under anaerobic conditions. So there are benefits to this. So, as I said, fermentation can produce CO2. It just depends on how many carbons are in your final organic terminal electron acceptor. If it's 2 or fewer, then those extra carbons from pyruvate need to go somewhere. We saw in ethanol fermentation that those 2 carbons escape as CO2. As humans, we're pretty intimately familiar with ethanol fermentation because we use it from bread making to wine production, beer production, and a lot of other fermented foods that we consume do make CO2 as a byproduct. And if you imagine that for every glucose you're making 2 carbon dioxides, that can really build up. So I just wanted to share this video with all of you. This is a barrel of wine that was overpressured. And he has just about uncorked it. So all of that pressure is from those 2 CO2s per glucose molecule. So I don't know about you, but I was quite impressed with that. Not all fermentations, as I discussed, lead to CO2 production. One of those is lactic acid fermentation. Lactic acid is a 3-carbon organic molecule, and so those pyruvic acids are just turned into lactic acid, and we don't have any consumption of CO2 there, or production of CO2. This table is a summary of what we've discussed so far. The summary has each step and the constituents we need to follow here, the ATP, the GTP, NADH, FADH2, and the CO2 produced, and where they are being produced at. In glycolysis, we saw we have 2 ATP and 2 NADH generated, and this is done in the cytoplasm. For pyruvate oxidation, we have 2 NADH produced and 2 CO2 produced as well. The Krebs cycle, we saw 2 GTP produced, 6 NADH, 2 FADH2s, and 4 CO2s. So if we add up all the CO2s there, we get 2 plus 4 equals 6, which would be equal to 1 6-molecule sugar molecule, glucose molecule. In the electron transport chain, we produce by far and away the most ATP at 34. We lose 10 NADHs. In other words, we generate our NAD+. We lose 2 FADH2s, and that's also done in the mitochondria. So the some processes we see on the next 3 lines are aerobic respiration produces 38 of a combined ATP and GTP. and we produce a sum total of 6 CO2 per glucose molecule. In ethanol fermentation, since we only get glycolysis, we only get to sum total of ATP, and we produce 2 CO2 and our 2 ethanol. In lactic acid fermentation, we get our 2 ATP from glycolysis and no production of CO2 because all of it is converted into lactic acid, a 3-carbon molecule, 2 3-carbon molecules. I recommend that you take this, this table and you study it very well. You might want to make a 3x5 note card out of this and just memorize these values.
Checkpoint 3. List the total combined ATP and GTP created from aerobic respiration and how much from lactic acid fermentation.
This table here shows the different types of fermentation. So there's quite a bit. We can have lactic acid fermentation, makes lactic acid. It's used in the processes of sauerkraut, yogurt, and cheese making. We have alcoholic fermentation for ethanol and CO2 as the products, and that is used for beer, bread, wine, and spirits. Mixed acid fermentation can create acetic acid, succinic acid, a variety of acids like that. Ethanol, CO2, and H2. It's used in processes such as making vinegar. Vinegar's main acid is acetic acid. Propionic fermentation produces propionic acid, acetic acid, and CO2. So if you've ever looked at the eyes of Swiss cheese, those eyes are bubbles made from CO2 production and propionic acid. Next, we have butane-diol fermentation, which makes butane-diol and CO2. This is used in producing Chardonnay wine, which gives that characteristic Chardonnay flavor. And finally, we have butyric acid, butyric fermentation, which makes butyric acid, H2, and CO2. And butyric acid, as its name is derived, has a buttery flavor to it. As seen in that table, lactic acid fermentation has an end product of lactic acid. It uses a bacterium called Lactobacillus, is the name of the genus, and there can be a variety of different species that are involved. If you ever look at your yogurt container, it probably lists 2 or more different bacterium used. It's used in a lot of products such as yogurt, pickles, sauerkraut, and it's also an important member of our gut microbiomes. Alcohol fermentation, the majority of which is done with Saccharomyces cerevisiae, that is a sugar-eating fungus that produces beer, as we remember what that name means. Its end product is ethanol and CO2. It's important for beer, wine, and spirits, but it's also used in biofuel production, as ethanol is a common biofuel, and it is a member of our gut microbiome. In fact, if you watch the news, you'll see this come up quite a bit. It's called autobrewery syndrome, where there's an imbalance in the gut microbiome and an overgrowth of yeast, which leads to an overproduction of ethanol and can cause people to endogenously produce ethanol and get them intoxicated. So you see issues with people getting into car accidents and things like that, not due to consuming alcohol, but rather producing alcohol. Scientists have ways for determining what sort of fermentation and if fermentation is being accomplished. This is done by using something called an inverted Durham tube in the presence of a pH indicator. So the pH indicator changes from red if there is no acid to a yellow if there is an acid being produced. So if you see anything that ends in ic acid or ate, ATE, that is something that is capable of being an acid. So propionic acid, lactic acid, acetic acid, acetate, propionate, lactate. All of those are just names for acids. So review that list, that table, with all of those those types of fermentations, and if they end in the word ate or ic acid, then that is something that is an acetic fermentation process. If an acid is produced, it will change the color of that pH indicator from red to yellow again. And what the inverted Durham tube is, it's just a glass tube that's turned upside down inside of the larger tube and filled with the solution of the medium. And so if you set this to incubate overnight, or however long you incubate the sample for is necessary, the air bubbles that don't go through the inner tube, that are produced from organisms that are not inside of it, the air bubbles typically will just escape out the top, diffuse through the solution and out of the tube. However, since you put this inverted tube here, you have a way to capture some of that gas. Some of that gas will rise up and hit the top of this inner tube and get stuck, and it will show up as a gas bubble. If you have a gas bubble, that's indicative of CO2 production. So this measures two different things. It can measure acidic fermentation. So ethanol, for example, is not an acid. It doesn't end in ate or ic acid. And if that acidic fermentation or non-acidic fermentation renders CO2 in the process. Does it have CO2 production? So here is an image of a variety of fermentation test outcomes. In figure A, you'll note that there is no acid because it remained yellow or remained red after fermentation, and there is no gas. If you look at this tube, you can see it has kind of a pinkish color to it, right? But if you look at B, it has gas, a gas bubble, because this is much more clear. I know it's a little hard to eye, but with a little practice, especially a side-by-side image here, you can see that this has a pinkish hue to it, and this looks much more clear. You can also have no gas and acid production. So as we can see, there's no bubble that rose up through the Durham tube, but there was an acid production here. And then finally, D, we have gas and acid. So here's the yellow pH indicator and a clear bubble at the top, as you can compare it to the other tube, which is just solid yellow.
Checkpoint 4. Which outcome would result from the culturing of a microbe from the Lactobacillus genus?
Currently, there is a large ecosystem of industrial microbiology companies that produce valuable products from the processes of fermentation. And this just requires some metabolic engineering, introducing of new genes to redirect that carbon to making other products, such as rose oil from yeast, or a sort of a spider silk. And I would like to revisit our metabolic classifications. So we have discussed a lot today about chemoheterotrophs, which get their energy source and their carbon sources from chemicals, from organic chemicals. So that was what happened during the process of cellular respiration and fermentation. Both carbon and chemicals or energy was derived from glucose. And most bacteria prefer to do this, most species of bacteria, and all of fungi and all of animals do this in order to obtain their energy and carbon sources. I am just going to talk very briefly about photoautotrophs, because without photoautotrophs, most of our organic matter just would not be available for use that we have here on planet Earth. That's because most of our energy comes from the sun, and it's used to convert inorganic carbon into our organic carbon. It's done through a process called photosynthesis, which uses the sunlight energy in order to synthesize organic compounds and obtain energy. And so this is performed by our photoautotrophs. And we have our algae, our cyanobacteria, some protozoa, and then of course a little bit higher on the scale is we have trees and things like that. It converts, again, sunlight energy and CO2 into chemical energy or food, carbohydrates, cellulose, things like that. And here we have an example of all three of those. We have our algae here on the bottom right, and algae tend to have a very square-like structure. That's because of the cellulose that's inside of algae provides a very rigid and strong cell wall. Here we have some protozoa. Protozoa tend to be very oblong. They tend to be active movers, and so they have kind of often this sort of aerodynamic shape to them. And here we have cyanobacteria. Obviously, we can see this one in the middle is going through the process of binary fission with its septum forming here, and it's going to form that Z ring, which is going to build a cell wall in between the two cells and eventually pinch off into a new cell. This looks to be sort of a bacillococci shape. So I'm not going to cover photosynthesis very much, but I am going to require you to at least know the reaction of photosynthesis. Photosynthesis, like we said, it takes light energy and it synthesizes organic compounds from inorganic compounds. And this is the classic example of that, where we take carbon dioxide and we need some water in order to get our hydrogens and our oxygens, our extra oxygens, and we produce a glucose compound and oxygen gas. This has a certain what we call stoichiometry or ratio of atoms and molecules, and that's because this equation needs to be balanced. We need the same number of carbons on one side as the other, because as we know in science, matter and energy is neither created nor destroyed. So we have to have some accounting of where the carbon is going and where it comes from, the hydrogen and the oxygen. And so I will recite that equation for you. It's 6 CO2s plus 6 H2Os creates 1 glucose and 6 oxygens. And if we count up all of the atoms, let's do that really quick. On the left side of the equation, we have 6 CO2s, or 6 carbon dioxides, which means we have 6 carbons. There are no carbons in water. And on the right side of the equation, there are no carbons in oxygen, but we see that there are 6 carbons in glucose. Let's follow up on our, let's do our hydrogen next. There are no hydrogens in CO2. There are 2 in water, and there's 6 water molecules. So 6 times 2 is 12. If we look to the right side of the equation, we see there are no hydrogens in oxygen, and there are 12 hydrogens in glucose. So we're balanced for hydrogen, and let's finally do oxygen. So we have 2 oxygens per carbon dioxide, hence di and dioxide, and we have 6 of them. So 6 times 2 is 12. We have 1 oxygen in water, so 6 times 1 is 6, and 6 plus 12 is 18. We look over to the right side of the equation, and we have 6 oxygens in glucose, and 6 times 2 for oxygen is 12. 12 plus 6 is 18. So there's 18 oxygens on both sides. It's important for you, and you need to expect that you'll have to recite this equation. There are some ways to help you remember, and that is if You know the four components. You know that there's carbon dioxide, water, and then glucose and oxygen. If you can remember one half of the equation, you should be able to calculate how many molecules you have on the other half. So for example, if we say that there are 6 CO2s and 6 H2Os, we know that there can only be one glucose because we have a 6 carbon molecule in glucose, which would balance out the carbon dioxide. Since we know there's one glucose, we know that we need to get our other 12 oxygens from the number of oxygen molecules. So since oxygen, atmospheric oxygen is O2, we would have 6 O2s. It does pay to memorize that whole equation. Number one, you're going to be a lot faster for taking an exam, but not all photosynthesis equations might look the same. You might have different products here at the end. And I do highly recommend just memorizing this whole equation. It's really not a lot. That brings us to the final checkpoint, checkpoint 5. Please write the balanced equation for photosynthesis. And now, just as we did for glycolysis, pyruvate oxidation, the Krebs cycle, and the electron transport chain, I want to discuss the stages of photosynthesis. I'm not going to ask you to remember everything that occurs in photosynthesis. It's a pretty complex process, but there should be some basic understanding here of where things are coming and going. For example, this process occurs in 2 separate stages. One is the energy harvesting stage, and this is where we take light and it goes through these photosystems, and these photosystems have an electron transport chain. So this is kind of similar to what we saw with cellular respiration, right? To harvest energy in the form of ATP, we had an electron transport chain. In photosynthesis, we have an electron transport chain to harvest ATP from water and sunlight here. So we create ATP, and we also get some NADPH out of this process. And then in the Calvin cycle, we can consume our ATP and our NADPH. pH is kind of like how we said GTP is like ATP. Well, NADPH is the photosynthesis version of NADH. We can use this to produce a variety of other sugars that can come out of this. And so these are the organic compounds that are used by fixing our CO2. And this is the second stage of photosynthesis, where we take our energy and our electrons and we use that to build other more complex compounds such as glucose, to be used in cellular respiration, cellulose, starch, other organic compounds. In the first stage, we use light and water, and we produce our oxygen. In the second stage, CO2 is consumed, ATP and NADPH is consumed from photosynthesis, and NADP plus and ADP are generated from it. All right, and one last note is that these occur in 2 different parts of the cell. So in algae, there are these chloroplasts, and these are like the mitochondrion. They are thought to be derived from ancient bacteria. In this situation, ancient cyanobacteria. Cyanobacteria are bacteria capable of photosynthesis. And it occurs on this inner membrane, it occurs on the thylakoid membranes, which are the specialized membrane system in chloroplasts. The Calvin cycle occurs in what is called the stroma of the chloroplast. So we have thylakoid membranes for our electron transport chain and the stroma for our Calvin cycle. All right, so this led to what we call the great oxygenation event. So when aerobic oxygenic photosynthesis evolved, that is photosynthesis that produced oxygen, it was first thought to be carried out by what we call cyanobacteria. This created a huge change in the makeup, chemical makeup of our atmosphere. And it happened about 2.5 billion years ago. And this, as you can imagine, created a massive extinction event because microbes weren't evolved to handle that oxygen and the reactive oxygen species it carries, and it allowed a flourishing of other microbes that kind of took over a lot of these environments and caused a mass extinction event. So that is the end of our discussion of microbial metabolism. Thank you very much, and we'll see you in the next presentation.